In this episode, we'll start by looking at a diagram of a microscope, then take a look at the different types of microscopes and learn their key differences, explore an important equation that allows us to calculate magnification, and finally, look at what standard form is and why we use it in science. Scientists first came to see individual cells with the help of light microscopes. The main components of a light microscope include the eyepiece through which a person looks through, objective lenses which are involved in refracting the light to magnify the image, a light source which amplifies the light entering the objective lens, a stage which holds the sample or specimen, a coarse focus adjuster for big shifts in focus, and a fine focus adjuster for smaller shifts in focus. And finally, an arm and a base for the microscope. Microscopes work by showing large images of very small samples or specimens. They magnify small objects with the help of light and lenses. However, the ability to see smaller subcellular structures such as ribosomes and plasmids was not possible with light microscopes. This was made possible with the help of more powerful and complex microscopes, known as electron microscopes, which were developed in the modern age. Whereas light microscopes use light to magnify an image, electron microscopes use electrons. These electron microscopes provide greater magnification and resolution. Resolution is the ability of microscopes to distinguish between two points next to each other. You will also need to understand how to calculate magnification. Magnification is calculated by dividing the image size by actual sample size. Additionally, you can also calculate any of the other components from this equation if you have the values for the other two components. Remember to make sure the units you use for the values are consistent. Convert any before calculating if required. As you can see, magnification is a very big number. Science has a way of dealing with very big or very small numbers, and no, it's not via the use of force. In science, we use something called standard form. This simplifies and expresses a long sequence of numbers in a very short form by shifting the decimal place. The first number in standard form is a number between 1 and 10, followed by a decimal place, followed by one or two other numbers found in the original value. This is then followed by a multiplication by 10 to the power of either a positive number if it's a very large number or a negative number if it's a very small number. The power of the number dictates the number of times the decimal point moves left for small numbers or right for large numbers.